and welcome everyone to today's webinar, Illumina DNA PCR-Free Prep Tagmentation and Introduction. Today we will be covering the following topics: advantages of Illumina DNA PCR-Free Prep, required consumables and equipment, input and workflows, the library prep protocol in detail, library evaluation and quality control, sequencing, analysis, and then we'll wrap up with some resources. So first, let's discuss the advantages of the Illumina DNA PCR-Free Prep. The Illumina DNA PCR-Free Library Prep is based on the Nextera bead-linked transposome, or BLT, technology. In this workflow, double-stranded DNA is added to the BLT-PF, the bead-linked transposome, PCR-free, which simultaneously fragments and adds partial adapter sequences to the DNA. After this, indexes are ligated onto the fragments, and the final library is eluted from the beads, ready for sequencing without any additional PCR amplification. There are several key features and advantages to the Illumina DNA PCR-Free Library Prep. First, it's a significantly faster workflow with only 55 minutes of hands-on time and 90 minutes of total turnaround time. It supports many DNA input types, such as genomic DNA, whole blood, dried blood spots, and saliva. We can expect final normalized libraries if we input greater than or equal to 300 nanograms DNA, or if we're using the integrated protocols for blood, dried blood spots, and saliva. There are also flexible protocol options. For example, with a thermocycler, we can either use standard input or low input and have two different workflows there. And we also have a workflow with the HiVex that supports standard input. This library prep kit is part of a comprehensive solution which includes the use of unique dual indexes, a whole workflow from sample to analysis, results interpretation with the TrueSight software suite, and is compatible with automation methods. Overall, the Illumina DNA PCR-Free Prep is part of a comprehensive workflow from library preparation to sequencing on most Illumina platforms, with several analysis options such as the TrueSight software suite and BaseSpace sequencing hub apps. Illumina DNA PCR-Free library prep reagents are automation-friendly, with scripts in development or already developed from these vendors. Please contact these vendors for more information about automating this library prep. Now we will review the required consumables and equipment. There are several components that need to be ordered before you're beginning your library prep. First, we'll need the library prep reagent kit itself. We have two options: one kit that supports preparation of 24 samples, and a second kit which supports preparation of 96 samples. We will also need at least one index adapter set, of which we have the IDT for Illumina DNA/RNA UD indexes, set A or set B, with set C and D coming soon. For most instruments and consumables, we will also need one of these custom sequencing primer kits. Either the kit that contains the Read1 VP10 custom primer, or the kit that contains both the Read1 and Index 2 primers. We'll go over this in much more detail in the sequencing section about which of these kits you'll need and how to choose them. And lastly, you will need the Illumina Lysis reagent kit if processing blood or saliva samples. There are also user-supplied equipment and consumables that should be acquired in advance. For the thermocycler workflow, we'll need a thermocycler. For the HiVex workflow, we'll need the HiVex system as well as a plate shaker. All workflows will require a magnetic stand, vortex mixer, and centrifuge. We will also need Kappa qPCR kit or single-stranded qubit reagents for quantification of the final libraries. Please consult the consumable and equipment section of the reference guide for a full list for your selected workflow and input type. Lastly, I would like to point out a few reagents that are unique to this library prep kit. So first are the single-stranded qubit reagents. Libraries generated from this library workflow are single-stranded and therefore cannot be quantified by standard double-stranded DNA qubit reagents. Please note that qPCR quantification is always a valid alternative. This prep kit also uses the Illumina purification beads. These beads are used for DNA purification and/or size selection of the DNA fragments based on a ratio of beads to sample. These beads are stored at room temperature, and freezing will damage their function. And then lastly, this kit uses the bead-linked transposome PCR-Free, the BLTPS. This is a unique BLT for this workflow, and it's also the only bead that Illumina manufactures that is stored at -20 in the freezer. These beads have been validated for up to 6 freeze-thaw cycles. Now we will review the input and workflow options for this library prep kit. Illumina DNA PCR-Free supports 4 types of input: genomic DNA, whole blood, dried blood spots, and saliva. For genomic DNA input, we will need high-quality genomic DNA. When we use 25 to 299 nanograms of input, we will be below the saturation point of the bead. Therefore, quantification of the input is required, and final libraries will not be normalized. When our input is between 300 and 2,000 nanograms, we will be saturating the BLTPS, so quantification of the input is not required, and final library normalization is expected. There are several resources for the genomic DNA workflow. First, the protocol can be found in the Illumina DNA PCR-Free reference guide. We also have a support page which contains several FAQ questions. And we have a web-based training which includes the product overview and a brief review of the protocol. For whole blood input, we need 20 microliters of fresh whole blood in EDTA collection tubes. These must be stored at 4 degrees Celsius and processed within 3 days according to the manufacturer's instructions. Processing of whole blood will require the Illumina Lysis reagent kit. And with whole blood, we expect to extract enough DNA to saturate the BLTPS, so high yield is expected. This means input quantification is not required and final library normalization is expected. The resources for the blood lysis workflow include, again, the library prep reference guide, where the specific blood lysis protocol is referenced in Appendix A. We also have questions on the support page FAQ. And the web-based training includes best practices for blood collection and storage, as well as blood extraction. For dried blood spots, we will need 6 × 3 millimeter squared dried blood spot punches and Whatman 903 protein favor cards. These should be stored at room temperature. Processing of the dried blood spots will require the Illumina Lysis reagent kit. Again, we expect to extract enough DNA to saturate the BLTPS, so high yield is expected. This means input quantification is not required and final library normalization is expected. The resources for the dried blood spot lysis workflow include the blood lysis protocol, which again is in Protocol A of the Appendix A of the reference guide. There is a support page FAQ. And the web-based training includes best practices on blood collection and storage and blood extraction. For the saliva workflow, we will need 50 microliters of saliva from Origin tubes. These will need to be heat stabilized for 1 hour at 50 degrees Celsius prior to storage at room temperature. Please follow the manufacturer's instructions for more details. Processing of saliva will require the Illumina Lysis reagent kit. Specifically, we will need the extra Illumina purification beads that's provided in this kit. We expect to extract enough DNA to saturate the BLTPS once again, so high yield is expected. so input quantification is not required and final library normalization is expected. Resources for saliva workflow include the Saliva Lysis protocol, again in Appendix A of the Library Prep Reference Guide. We have the support page, FAQ, and the web-based training includes Saliva Lysis best practices. So depending on our input amount, we will be following different workflows, and we may or may not expect normalization of the final library. For instance, if our input is 25 to 99 nanograms, this is considered low input. 100 to 299 nanograms of DNA is considered standard input, but it will be below the saturation point of the BLTPF. 300 to 2,000 nanograms is considered standard input, and it will saturate the BLTPF. And then blood and saliva are considered blood and saliva input. When we are below the saturation point of the BLTPF, then we will need to quantify our input. So anything in the 25 to 299 nanogram range. When we do saturate those BLTPF, then we can expect final library normalization to occur. And then lastly, we have different protocols and workflows depending on these. So the 25 to 99 nanogram DNA of low input has its own workflow, the thermocycler low input protocol. 100 to 299 nanograms of DNA will be following the thermocycler standard input protocol. When we have 300 to 2,000 nanograms, we can either follow the thermocycler standard input protocol, or we can follow the HIVEX protocol. And then lastly, the blood and saliva, once we've extracted the DNA, will follow the thermocycler standard input protocol. And please note, for DNA input quantification, we should always quantify with a fluorometric method, such as the qubit double-stranded DNA high-sensitivity kit. So we have separate chapters in the reference guides for the various workflows that can be employed. The choice in the workflow will depend on our input amount, input type, and equipment usage. For example, the thermocycler standard input will be used if our input is between 100 and 2,000 nanograms. Again, the amount we use will determine whether or not we saturate and if the final libraries are normalized. The thermocycler standard input, again, will support whole blood, dried blood spots, saliva, in addition to the genomic DNA. And the steps for the thermocycler standard input can be found in Chapter 2 of the reference guide. The thermocycler low input workflow, which again supports 25 to 99 nanograms of genomic DNA, can be found in Chapter 3 of the reference guide. And finally, the HIVEX workflow, which supports greater than or equal to 300 nanograms of genomic DNA. This will be found in Chapter 4 of the reference guide. To ensure success during library preparation, our input must be clean and pure. Purity can be assessed with a UV absorbance method like a nanodrop. Samples should also be free of contaminants such as EDTA, phenol, ethanol, and other organic contaminants, which are known to inhibit the tagmentation reaction. So now we will review the library prep protocol in more detail. So in general, for libraries to sequence on Illumina instruments, the DNA insert needs to be flanked with sequencing adapters. Each adapter has 3 parts. First, for clustering, libraries must have a P5 and P7 binding region at the far end of the library. For sequencing, the libraries must have sequencing primer binding regions, specifically the READ1 sequencing primer binding site in orange, and the READ2 sequencing primer binding site in blue, so that we can sequence the DNA insert. And lastly, in order to mix multiple samples in a single run, libraries must have a unique index or barcode sequence, shown here as Index 1 and Index 2. Please note that for the Illumina DNA PCR-Free library prep, we're generating single-stranded final libraries. Before beginning your Illumina DNA PCR-Free library prep, please ensure to prepare the consumables in advance. Prepare diluted HP3 or sodium hydroxide and 80% ethanol for each reaction. If following the Hybex protocol, confirm the Hybex with the MIDI insert is preheated to 45 degrees Celsius. If following the thermocycler protocol, please confirm the TAG and ELM programs on the thermocycler are pre-programmed. So here are the steps involved in the Illumina DNA PCR-Free Library Prep protocol. First, we're going to Tagment Genomic DNA. We will perform post-Tagmentation cleanup. We will Ligate Indexes and clean up the libraries, at which point we'll reach a safe stopping point. When we're ready to sequence, we can quantify and pool the libraries, dilute to our starting concentration, and set up our sequencing run. No matter the source of input DNA, all input types will go through the same workflow as shown on the right. When using one of the workflows for blood or saliva, please note that extracting the DNA via the protocol found in Appendix A, the isolated DNA will be fed into Chapter 2 for preparation with the thermocycler standard input workflow, and then follow the same downstream steps. So the first step of the Illumina DNA PCR-Free Prep protocol is to fragment and tag the DNA with partial adapters, a process called Tagmentation. First, we will prepare the Tagmentation reagent, the bead-linked transposome PCR-Free, BLTPS, and the Tagmentation buffer, TB1. We'll incubate the enzymatic reaction, which is 50 microliters of the DNA in the Tagmentation reagent, and run the TAG thermocycler program or incubate on the Hybex. The best practices for this step include following the protocol as written, using the preparation steps in the section to prepare all reagents in advance. When handling the BLTPS, bring this reagent to room temperature and vortex fully before use. After use, it should be stored frozen and upright to make sure that the beads are submerged in the buffer. And when using this reagent, make sure to aspirate and dispense slowly due to the high viscosity of the solution. In the second step, post-Tagmentation cleanup, we will stop the Tagmentation reaction and wash the DNA. We will add the Stop Tagment Buffer 2, ST2, and incubate at room temperature. We will then wash with the Tagmentation wash buffer. We will incubate, leaving the supernatant in the plate to prevent the beads from overdrying, and then place on a magnet. The best practices for this step are to dispense ST2 directly onto the beads, pipette TWB and ST2 slowly to minimize foaming, and to fully resuspend the beads with the Tagment wash buffer. In the next step, we will ligate the I5 and I7 index adapters to each sample. First, we will add the extension ligation mix, and then we will add the indexes, which are the IDT for Illumina DNA RNA UD indexes, either set A, B, C, or D. We will run the ELM thermocycler program or incubate on the Hybex. We will then perform a second wash with the Tagment wash buffer and elute with diluted HP3, which is sodium hydroxide, which results in single-stranded libraries being eluted. The best practices for this step is to make sure all the Tagment wash buffer has been removed from the samples before adding ELM and adapters. Aspirate and dispense ELM slowly due to the viscosity of the solution. For the index adapter plate, each well of the plate is single-use only, and you should puncture the foil seal prior to use with a pipette tip. For indexing, we'll be using the IDT for Illumina DNA/RNA UD or Unique Dual Indexes. These indexes come in a plate format where each well contains a unique pair of an I5 and I7 index combination. Each well is single-use only. For low-plex pooling, please consult the index adapter's pooling guide. These indexes will be pooled by column rather than by row. So for example, if we only have 2 samples, we will use the first 2 or the last 2 wells in a column. If we have 8 samples, we will use an entire column. Please ensure that sample sheets include 10 cycles of sequencing per index read, since these are dual 10-base pair indexes. In the next step, we will size select and clean up the libraries using Illumina purification beads. When we have 25 to 99 nanograms of input, please note that it will be a single-sided size selection to maximize the library yield, whereas if we have 100 or more nanograms, we will be using a double-sided selection to more tightly regulate the library size. After we collect the supernatant from the adapter ligation, we bind the DNA to the Illumina purification bead. Then we will wash the beads with 2 rounds of ethanol wash, and then elute the libraries in resuspension buffer. The best practices for this section revolve the Illumina purification beads. These should be brought to room temperature before use and fully resuspended by vortexing frequently. Make sure to use freshly prepared 80% ethanol for the washes, and ensure that the correct bead volume is used for each size selection step, as the volume will affect the size of the library that is captured. At this point, our libraries are complete and ready for ready for library evaluation and quality control. When assessing the library's quality, please note that the Illumina DNA PCR-free libraries are single-stranded due to the elution of sodium hydroxide. Due to this, the libraries cannot be visualized on a bioanalyzer, tape station, or fragment analyzer. Therefore, we will use 450 base pairs as the library size for molarity calculation purposes. We do have a tech note linked here, the tunable insert sizes of Illumina DNA PCR-Free Prep Tagmentation, which reviews how to modulate that insert size between 350 and 550 base pairs. Only if you're following this modified workflow should you change that 450 base pairs to the target size that you're using when calculating molarity. We have 2 methods for quantifying Illumina DNA PCR-free libraries. When the input is greater than or equal to 300 nanograms, we can quantify with a single-stranded qubit reagent. Please note that the use of double-stranded qubit reagents will result in a value of 0 nanograms per microliter. When calculating the molarity, we will use the following equation, which is based on a 450 base pair size and 660 gram per mole DNA mass. This reduces to the molarity equaling the yield in nanograms per microliter times 3.36. The output of this equation is a double-stranded DNA molarity equivalent. We can also quantify libraries by kappa qPCR. This is required when the input is less than 300 nanograms, but it can be used for any workflow and input amount. When calculating molarity, please use 450 base pairs as the size and 660 grams per mole as usual for the DNA mass. This again will output the double-stranded DNA molarity equivalent. And please note that when the input is more than 300 nanograms, quantification of individual libraries is not required because we've reached the saturation point. We will only need to quantify the library pool. When you're ready to sequence, we will denature and dilute the libraries. If sequencing on a NovaSeq, guidance can be found in the reference guide, with loading concentrations provided for standard and XP workflows. Please note that we have different loading concentration values depending on if we use qPCR or single-stranded qubit to quantify our libraries. If following the low input workflow, we will only be able to sequence on the XP workflow due to the expected lower final library yield. If sequencing on the NextSeq 1000, 2000, we will use 1000 picomolar as our loading concentration if we're doing onboard denaturation. This detail can be found in the NextSeq 1000 and 2000, the sequencing system guide. If doing manual denaturation, then our loading concentration will be 100 picomolar. Guidance can be found in the NextSeq 1000 and 2000 Denature and Dilute Libraries guide. For all other supported instruments, please refer to the optimal loading concentrations for Illumina DNA PCR-Free Libraries tech note determine the appropriate loading concentration for your instrument. Please note that all workflows should still follow standard denature and dilute protocols where we add sodium hydroxide. This will remove secondary structures. Now our libraries are ready for sequencing. Illumina DNA PCR-Free libraries are compatible with most Illumina sequencers, such as the MiniSeq, MiSeq, NextSeq 500/550, NextSeq 1000/2000, HiSeq platforms, and NovaSeq 6000. Please note that the Illumina DNA PCR-Free libraries are not supported on the iSeq 100 or the HiSeq X. And note that most platforms and consumables will require a read 1 or a read 1 and index 2 custom sequencing primers, which we will review in a few more slides. When preparing for sequencing, we must take these 4 factors into consideration: the sample sheet, pooling guidelines, denaturing and diluting our libraries, and run setup. For the sample sheet, Illumina Experiment Manager version 1.19 has been updated to include this library type by default. We also have, for Local Run Manager, library prep kit definition files available for download. When pooling libraries, we need to consider the samples per run as well as low-plex pooling. To help determine the number of samples per run, please consult our data sheet, application notes, technical notes, and the sequencing coverage calculator. When doing low-plex pooling, please consult the Index Adapters Pooling Guide to know which indexes are the optimal ones to use for color balance. When denaturing and diluting the libraries, we should follow standard protocols to denature with sodium hydroxide. Again, we're going to add sodium hydroxide to these single-stranded libraries to help remove secondary structures. We will then dilute to the target loading concentration for the instrument. For run setup, in most cases, we will need to add the custom sequencing primer or primers to the cartridge. Therefore, we need to instruct the instrument to pull from these wells. So we will either select read 1 or read 1 and index 2 as needed in the control software. I will go over this in more detail in a few more slides. We'll also need to make sure the instrument is set up to the correct read length. And then on older MiSeqs that use only the sample sheet for run setup, we need to ensure that this sample sheet specifies a custom read 1 primer so that we use the custom read 1 primer during sequencing. So as promised, we'll now discuss the required custom sequencing primer options in more detail. Due to how different Illumina instruments sequence the index 2, some instruments only require a custom read 1 primer. Other instruments require both custom read 1 and custom index 2 primers. For this reason, we offer 2 custom primer kit configurations for purchase, which are sold separately from the library prep kit. So the first option here in purple is the read 1 primer. This kit will contain the VP10 custom read 1 primer. We will get 4 tubes of it, and each tube will have 7.5 mils. This kit is designed for use on the NovaSeq 6000 with version 1.0 sequencing reagents, MiSeq, HiSeq 1000, 1500, 2000, and 2500. The other kit contains the read 1 and index 2 primers. So specifically, it contains the VP10 custom read 1 primer. This has only 2 tubes of it at 7.5 mils each. And then we have the VP14 custom index 2 primer. Again, 2 mils of this at 10.5 mils each. This kit in blue is designed for the MiniSeq, the NextSeq series, the HiSeq 3000, and the HiSeq 4000. So again, please note that these kits are sold separately from the library prep kit as it depends on what instrument you're actually sequencing on or if you're sending it to a core. Custom primers are compatible with Illumina DNA PCR-Free, PHI-X, and other Illumina libraries. So essentially, these custom primers are a cocktail that will sequence all of these library types. The custom primers are provided at final concentration and require no dilution. Therefore, we will never spike these primers in. We will always use the dedicated custom primer well. And lastly, the custom primers are not required for the NovaSeq 6000 version 1.5 sequencing reagent. This newest SBS kit already contains the primers, and that's why it's the one exception to needing the custom sequencing primers. So we've created a resource to help clarify which primer to use, how much to use, and where to load it for each instrument in our bulletin, Custom Primer Requirements for the Illumina DNA PCR-Free Prep Tagmentation Kit bulletin. So this bulletin contains 2 tables to help you set up your run. It's going to be a bit overwhelming what I'm going to show you, but I'm going to walk you through a couple of examples. And I'm showing the full tables here so that you have them as a resource, but you can always consult the bulletin for full details. So in this first table I'm going to show, this table will provide guidance on which primer is required for instruments, the volume of the primer to use, and what well to load it in. So here's the table. In the leftmost column, we have the Illumina instrument. In the second column, we have exactly which of those 2 primer kits you should be ordering. The next 2 columns show how much of the VP10 read 1 primer to load and where to load it. And the final 2 columns show the VP14 Index 2 primer, how much to load and where to load it. So let's walk through a couple of examples. So if we have a MySeq, we will need the custom primer kit ending in the number 6, which only contains the read 1 primer. We will need to pipette 600 microliters of this read 1 primer into well position 18. The MySeq does not need an index 2, so we will leave that empty. For the next Seq 500 and 550, we will purchase the primer kit ending in the number 7. This contains both the read 1 and the index 2 primers. We will pipette 2 mils of the read 1 primer into position 7 and 2 mils of the index 2 primer into position 9. For the NovaSeq, using version 1.0 SBS, we will need to purchase the custom primer kit ending in the number 6. For SP, S1, and S2 flow cells, we will pipette 2 mils of the read 1 primer into position 5, whereas S4 flow cells will pipette 3.5 mils of the read 1 primer into position 5. This run setup does not require index 2 primers. And then the last example is the NovaSeq 6000 version 1.5 consumables. Again, this new kit of consumables already has the primer, so we don't need to use any custom primers. Again, please see the bulletin for full details or if you need this as a reference. So the second table in this bulletin is going to provide a guidance on how to tell the instrument to use those custom primers, and it also provides the recommended read length. So here's the table. So in the leftmost column, we have the software that is used to set up the instrument run. In the next column, we have the applicable instruments for that software. We then specify whether a sample sheet is required for run setup. Please note that a sample sheet is always required for demultiplexing, but since that can happen after a run, this column only specifies when it's needed for run setup itself. We then have a column about the library prep kit selection, a column for the custom primer run setup. So this is how we actually tell the instrument to use the custom primers. And then the final column is the read length of the run in the order of read 1, index 1, index 2, read 2. So again, we're going to go through a couple examples here to give you a feel for how to use this chart. So if we're sequencing on a MySeq with controlled software version 3, which is using Local Run Manager for run setup, we're going to set up our runs in Local Run Manager. We can download the library prep kit definition file and import it as a custom library, so this prep kit shows up in the drop-down. We will still need to tell the instrument to pull from that custom primer well rather than the Illumina well, so we will need to manually select custom read 1 during run setup. The read length for this run will be 151, 10, 10, 151. For the Mini-Seq or the NextSeq 500, 550, on the newer softwares of Windows 10, which again use Local Run Manager, we can download the library prep kit definition files and import them as custom libraries. Again, we need to instruct the instrument to pull from the custom wells instead of the Illumina wells, so we will need to manually select read 1 and index 2 as custom primers during run setup. The read lengths for the Mini-Seq and the NextSeq are 149, 10, 10, 149 due to the lower volume of overfill on these cartridges. For the NovaSeq 6000 with the version 1 consumable, we don't need a sample sheet for run setup, only for demultiplexing. And since we're just setting up the run in the control software, we're just entering in the read lengths and specifying custom primers. So again, we will need to manually select custom read 1 primer usage during run setup and set the read lengths to 151, 10, 10, 151. On the NovaSeq with the version 1.5 consumables, again, this cartridge has been updated to include these primers, so no selection of custom primers is required. And we will use the same read lengths, 151, 10, 10, 151. Again, this table can be found in the bulletin linked on the left. Now that our libraries are sequenced, we're ready for analysis. If data was generated using the NovaSeq, then we can use the TrueSight software suite for analysis and interpretation. Data from any instrument can also be analyzed in our BaseSpace Sequencing Hub app. The TrueSight software suite is designed for translating sequencing data into meaningful, interpretable results in rare disease cases. It can perform secondary analysis, including comprehensive and rapid variant calling, interpretation and reporting, case management, and it is an enterprise informatics with a holistic infrastructure and software suite. For more information, please contact our software and bioinformatics team within Illumina Technical Support. Illumina DNA PCR-free data can also be analyzed with BaseSpace apps. We have a variety of apps available, whether you sequence small, large, or human genomes. If you need help selecting an appropriate app, please contact our software and bioinformatics team within the Illumina Technical Support. Now I would like to provide you with some resources and links for the Illumina DNA PCR-Free Library Prep Kit. So first, there is the library prep protocol itself, which is the Illumina DNA PCR-Free Library Prep Tagmentation reference guide. We also have the data sheet for this library prep kit, and we have 4 technical and application notes that may be of interest. So the optimal loading concentrations for Illumina DNA PCR-Free libraries will have the loading concentration and primer requirements for non-NovaSeq instruments. The high-performance whole genome sequencing with Illumina DNA PCR-Free Prep Tagmentation note reviews the benefits of a PCR-free library and also has information on the number of human samples per flow cell on the NovaSeq. Tunable insert sizes with Illumina DNA PCR-Free Prep Tagmentation reviews how to modulate the insert size between 350 and 550 base pairs by adjusting the bead cleanup ratios. And the microbial whole genome sequencing with Illumina DNA PCR-Free Prep Tagmentation reviews how to sequence microbial libraries with varying GC content with the Illumina DNA PCR-Free Prep Kit and the results to expect. Here's a link to our support page for this library prep kit. Here's a link to the Local Run Manager library prep kit definition files to download and import into Local Run Manager for run setup. And lastly, here's a link to the Illumina DNA PCR-Free online training. Here are links to the system guides for instruments that support the Illumina DNA PCR-Free library. Again, in your slide deck, these will all be clickable links. And here are links to the custom primer guides and the Denature and Dilute guides for the instruments that support Illumina DNA PCR-Free libraries. And we have a couple of additional resources. So for indexing, we have links to the adapter sequences document, which has all the sequences for Illumina adapters. The index adapter pooling guide has both the plate layouts of the index kits, and it also says which indexes to use if combining fewer than 12 samples in a run. The index sequencing overview guide reviews how different instruments sequence. This explains why some instruments need just a custom read 1 primer and why other instruments need a read 1 and an index 2 primer. We also have several bulletins that we've created that are related to the Illumina DNA PCR-Free library. So the first one is the one that we reviewed a bit in those 2 slides. It's the custom primer requirements for Illumina DNA PCR-Free Prep Tagmentation kit. We also have bulletins on the bead types and Illumina library preparation kits, Illumina library prep kits and associated index kits, the Illumina adapter portfolio, and what sequences do I use for adapter trimming. We also have an upcoming bulletin on the considerations for choosing an Illumina DNA PCR-Free workflow, which again reviews exactly what equipment and consumables and other considerations you need for your input type and workflow. Here's a link to the sequencing coverage calculator to determine how many samples per run for whole genome sequencing.